Hello everyone, and welcome to the video lecture on neurology. I'm Stephen Alsop. You will notice that in the book, this chapter is composed differently from the others. Anatomy and physiology are combined into a single section. As you'll see during the course of this lecture, this is a direct result of the fact that anatomy and physiology in the nervous system are very closely linked. There are around 86 billion neurons in the brain, with even more connections. The entire purpose of the brain is to allow us to take appropriate actions based on external and internal stimuli. Thus, we have a sensory system which processes stimuli and a motor system that engages action. And now, this is certainly a reductionist way of viewing it, but in a sense, all the other parts of the brain are dedicated to how to best utilize sensory information to produce adaptive responses. Now, before we delve into how the brain achieves this and how things can go wrong, we have to understand something about how the brain is actually put together. Because as I said, how the brain is connected dictates the manner in which it functions. So let's start at the very beginning. The early embryo is composed of three primary germ layers: the ectoderm, the mesoderm, and the endoderm. Now, these layers are covered in more detail in the reproductive chapter, and you should be aware of the adult derivatives from each structure. However, for the purposes of the nervous system, we are really interested in the ectoderm and the mesoderm. Now, the mesoderm will differentiate into the notochord. The notochord is a ventral structure that, among other things, secretes a signaling molecule known as Sonic the Hedgehog, and not the video game. Now, this molecule travels dorsally, and it's inhibited by another molecule which is called Wnt. Now, Wnt travels ventrally. So this signaling and inhibition is responsible for setting up the dorsal and ventral axes of the embryo. Now, under the signaling influences of the notochord, the ectoderm goes on to become the neural tube. Now, it is really amazing to think that our entire nervous system starts as this hollow tube and eventually develops into the most complex organ in our bodies. If you look at the diagram, you will see that around day 21 in the formation of this tube, there are a few cells that remain outside of the tube. These are the neural crest cells. These will go on to differentiate into a wide variety of cell types. You should be familiar with all of the cell types that neural crest cells can develop into. Now, a few of them include Schwann cells, you can have melanocytes, you can also have enterochromaffin cells. Excuse my handwriting, I'm practicing to be a doctor. You will also notice that even at this early stage, we begin to see the emergence of asymmetry with a dorsal and ventral positioning that will later dictate the development of the sensory and motor systems. The neural tube continues to develop, and by week 4, there is already a sense of differentiation between what will eventually become very different functional parts of the brain. You should definitely take the time to memorize the name of each of the primary vesicles shown here. By week 5, these primary vesicles have further developed into secondary vesicles. It is most important to know which of the vesicles become which regions of the adult brain. The way this is likely to be tested is by giving you a question about a neurodevelopmental condition that leads to a defect in the development of some specific brain region. And then they'll ask which primary or secondary vesicle was involved. For instance, a defect in Hox signaling in the prosencephalon can lead to what type of disorder? The answer is holoprosencephaly. This disorder occurs when the prosencephalon fails to develop into the two lobes of the telencephalon that later become the cerebral hemispheres. It is helpful to think about the development of these brain regions in a functional manner. For instance, the parts of the brain that will become the cortex are separate from the midbrain and hindbrain. The forebrain is then further subdivided because the cortex is divided into cortical structures such as the motor cortex, which would be roughly around there, and the subcortical structures such as the hypothalamus, which is around here, and the thalamus, which would be around there. Now, the brain and spinal cord make up the central nervous system,
while all other nerves and ganglia outside of the brain and spinal cord make up the peripheral nervous system. Let's try to understand the origin of this dichotomy. The important things to know are the embryonic layers from which the various parts of the nervous system develop. Remember that there are three primary germ layers: the ectoderm, the mesoderm, and the endoderm. Under the influence of various signaling molecules, part of the ectoderm will differentiate into the neuroectoderm. The neuroectoderm will then become the neural tube. In this process, there are a few cells known as neural crest cells that will become separated from the tube. These cells will develop into the peripheral nervous system. Meanwhile, most cells in the central nervous system will come from the neural tube. There is one exception to this rule, and that is microglia, which originate from the mesoderm. As I mentioned, the neural tube eventually forms the nervous system. Therefore, it stands to reason that defects in neural tube development can lead to very serious problems. Here we see a T2 weighted fetal MRI from both a sagittal as well as dorsal view. It shows a neural tube defect consistent with a meningomyelocele. Remember that T2 images will enhance water, and so in this case CSF will be brighter than its surroundings, as seen here. One important thing to remember is how neural tube defects can be detected. These defects are usually diagnosed prenatally through amniotic fluid screening. So what are the two proteins that can be measured as surrogates of neural tube defects? The answer is acetylcholinesterase as well as alpha-fetoprotein. Their protein levels would be elevated in the amniotic fluid. However, this is not a perfect measure, as AFP levels can be normal in meningocele, and this is actually one important distinction between meningocele and meningomyelocele, and we'll talk about that in a brief moment. So, as you can see, there are varying degrees to which failure of the tube to close can lead to problems. But let's start with a normal view of the spinal cord in a transverse section, as seen here. Now, spina bifida disorders occur when there's a failure of the neural tube to close, which can lead to defects in the vertebral column. Spina bifida occulta is the mildest form of spina bifida. Although the vertebra does not close, there is no actual herniation of the spinal cord or the meninges. You should remember that spina bifida occulta can be recognized by a tuft of hair or dimple on the back at the level of the defect. This is usually actually a substantial clue for a question on the boards. Meningocele and myelomeningocele are more severe than spina bifida occulta, with myelomeningocele being the most severe defect. In meningocele, only the meninges herniate through the vertebral column, whereas in myelomeningocele, both the meninges as well as the spinal cord herniate through the column. Now, the trick to remembering the difference between these two conditions is in the name itself. Meningocele refers to the fact that only the meninges herniate, while meningomyelocele includes the additional term myelo, which refers to the spinal cord, letting you know that both the meninges as well as the spinal cord herniate in this condition. Spina bifida, as well as meningoceles and meningomyeloceles, all develop from failure of the posterior neural tube to close. But of course, this can happen at the other end as well, and that's what we'll talk about next.